Hello, everyone. Welcome to today's live broadcast, the use of Raman Spectroscopy in the pharmaceutical industry, from discovery to the warehouse. I'm Laura Bush, the editorial director of Spectroscopy, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by Spectroscopy and sponsored by Bruker Optics. Bruker Optics, part of the Bruker Corporation, is a leading manufacturer and worldwide supplier of Fourier transform infrared, near-infrared, and Raman spectrometers for various industries and applications. We have a few housekeeping announcements before we begin. The webcast is designed to be interactive, so we encourage you to ask questions during the event. You can submit your questions by typing them in the Q&A box, and you can find that by clicking on the red Q&A widget at the bottom of the presentation window. You can enlarge the slide window by clicking on the small green icon in the upper right-hand corner of your window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically. If you have any technical problems viewing or hearing the presentation, please click on the question mark Help widget in the dock at the bottom of your window. Please also note that there are various resources available for our attendees, which you can see on the screen. You can download these directly at any time during today's event. I would now like to introduce today's speaker. Thomas Tagg is the Applications Manager for FTIR and Raman products for Bruker Optics. He's a member of the advisory boards of the Metropolitan Museum of Art in New York, EOS Photonics, and the New York Task Force on Industry Higher Education Partnerships. Tom received his PhD in chemistry from the University of Utah and his BS in chemistry from the University of Texas at San Antonio. He is a member of the American Chemical Society, the Society for Applied Spectroscopy, and the Microscopy Society of America. Tom has more than 30 peer-reviewed publications and 5 U.S. patents. Tom, thank you for joining us today. Please go ahead and get us started.
Well, thank you, Laura. So I'm glad that you could join with us today to discuss Raman spectroscopy applications in the pharmaceutical industry. Today I'd like to discuss different applications where we're going to look to apply Raman spectroscopy early on from the discovery phase all the way out to the warehouse and doing quality control of products. You'll be able to see that we have a new product offering that we'll cover at the end that we think will be very innovative, and we also have other techniques that are very much of great value to the pharmaceutical community today and hopefully tomorrow. My colleague John Richmond is not going to be able to attend today. He had travel plans that came up unexpectedly. So I hope I can fill in adequately for John. We're going to cover a few things in the order as shown on the slide. So we're going to talk about Raman spectroscopy. Some people might not be familiar with what it is and what it can do for you. So we'll cover those items. And then we'll talk about Raman microscopy in the pharmaceutical world. And remember that with microscopy, yes, we're going to look at small things in that case. Then we'll talk about handheld Raman for quality control and high-throughput screening. And there are 2 new technologies that are novel to this product using sequentially shifted excitation Raman spectroscopy to remove or mitigate fluorescence interference. And then also we're going to use simultaneous 2-laser excitation, and we'll talk about that. And then last, we'll look at some good examples for high-throughput screening. And as Laura stated, if there are any questions, we should have time to get to all of them at the end. Okay, so first, the Raman effect was discovered by Professor Raman in 1928. And for trivia questions, he used the sunlight as his source and use his eye as the detector. So if you're ever on Jeopardy, those are the key answers to getting the double Jeopardy right. With that, what we're really looking at is when we have a molecule, in this case benzene, and we're going to shine intense light, and in modern spectroscopy we're using laser light, we'll shine that light onto molecules, and we'll see what happens to the light. As one would expect predominantly the light passes through and does not interact with the benzene. The light can also bounce off elastically. But what we're more interested in from a spectroscopic point of view is the light that is either gained or lost from this interaction. So just a couple of basic things is that you can do the excitation from the UV all the way to the near infrared as shown in the first sentence there. And remember that this is not a strong effect, so only about 10 to the minus 8 to 10 to the minus 11th of the light is actually going to give us the desired Raman spectrum. Another important factor is that if there's an electronic transition that can occur with the excitation laser wavelength you're using, then we would have strong fluorescence interference that can sometimes dwarf the Raman signal. So what we're really interested in here in this slide with just reviewing the Raman principle is we're going to excite up from the ground state to the first excited state through a virtual state. As shown on the left, that gives us the information on what we call the Stokes shift, the 0 to 1 transition, which we're interested in. There's also the anti-Stokes where you start in the first excited state and decay down to the ground state, and the Rayleigh line or the zero line where there's no change in energy. So at this point, you can say, well, will any excitation laser wavelength work? And the answer is not really. As it turns out, there is usually some electronic transition that will be there, and it's just a question of where it is, and there can be multiple electronic transitions even accessible. In this case, if we just look at thioindigo, which with 488 excitation, you can see by the yellow spectrum there that, well, it just yields terrible fluorescence. But at 1064 excitation, you achieve and obtain a very nice Raman spectrum that can be easily interpreted and searched over a database. Okay, so why not do everything at longer wavelengths, say at 1064? As it turns out, well, the scattering efficiency falls off as the fourth power as you go to longer wavelengths. And in addition to that, you actually can't, the detectors just aren't as sensitive in the near infrared as they are in the visible. So you can see one of the schemes that works really well is to use more than one laser. So on the left, you can see if there's no fluorescence interference, just use the low wavelength high energy laser and you obtain a great result. If there is the electronic transition in the way, then you would just use a longer wavelength laser. And the opposite is true. Sometimes those the fluorescence interference can be at longer wavelength and you need to go to shorter wavelengths. And so having two lasers can be very important in obtaining a good Raman spectrum. So here's a slide that just shows that if the scattering efficiency does go as the fourth power, so if you can work at the shorter wavelengths, you're better off doing so. Okay, so what does Raman give to you? So the first thing that's not on the slide that is important to realize is that Raman spectroscopy and infrared spectroscopy in general give you very high levels of specificity. Each molecule has its own fingerprint signature in the infrared and Raman. And so with Raman and infrared, they're also intended to be non-destructive analysis tools. So now Raman versus infrared or near infrared, let's talk about that over the next couple of slides. For vibrational information, you're going to obtain really rich spectra. There are a lot of bands that will have transitions in Raman as well as in the infrared. Typically, you're looking at symmetric modes of vibration, so it's highly complementary, the Raman is, to the infrared. And so things that are weak in the IR are usually strong in Raman. So it's very helpful for carbon-carbon bonds, sulfur bonds, and so forth. There are also a wide range of applications that are available. So virtually any sample that you might look at in the infrared, it works great for Raman, with the lone exception being gases. The cross-section for Raman analysis is just too weak typically for a gas phase work. Maybe another point when we're talking about intensity is to realize that in the infrared you can do, your detection limits are actually lower than Raman, again because Raman is a weak effect. Well, when you look at doing things in water like frequently we do in the pharmaceutical world, it's really important to realize that water is a strong interferent in the infrared, but it's not at all a problem in Raman. And in fact, you can do things in aqueous solution in a very trivial manner. So Raman can be very useful for many applications where you want to do things with water. The bands are a little sharper in Raman than IR because of less nearest neighbor interactions. The other thing that you really get with Raman that sometimes is very challenging in the infrared is gaining access to what we call the terahertz region or the far infrared region of the spectrum. And with Raman, you're looking at the difference from the zero line. So getting down to 70 wave numbers is not a problem. You don't have to worry about the strong water absorption down in that region, and you don't have to change beam splitters or have other problems that you have to deal with in the infrared. But when you look at inorganics or organometallics and you want to look at polymorphs, the Raman is immensely useful for doing that. Next, and kind of in summarizing the Raman benefit, well, the nice thing about working with visible and near-infrared excitation is that you can use quartz and glass freely, where they're opaque in the infrared. And, for Raman spectroscopy, they're just not a problem at all. So it's very easy to get to samples. You can analyze things neat. You can analyze them in vials. You can have long fiber optic cables up to 100 meters long to get into the manufacturing environment, no problem. For quantification, because the bands are a little sharper, things work out a little easier for doing quantification. And maybe in summary, you have all the advantages and information of the mid and far infrared with the sampling benefits of the near infrared. Okay, so let's talk about the actual applications now. So we're going to go through some examples that will be important for the development cycle. We'll talk about metabolic monitoring of drug effects, and my favorite, reverse engineering of packaging and products, which obviously saves time over having to do the development yourself. And then quality control, where you're actually identifying unknowns, or doing compliance testing, where you're looking at raw materials and making sure that everything that is coming in is exactly as it should be. Well, one of the pitfalls historically of Raman spectroscopy has been wavelength accuracy. If you're going to look at metabolic changes in proteins, for example, you want to be able to have the wavelengths very precise on the as far as having the band positions right, and more importantly, even picking up small changes for differences in polymorphs or differences in protein structure. So with the Centera Raman microscope that we offer, we actually have a patent on doing the calibration internally. So we use a neon reference lamp that goes coaxially with the Raman signal on through the spectrometer and onto the CCD detector. And what this means is that now we use one part of the detector to do the Raman detection, and the second part of the detector to monitor the neon lamp, so that we can continually be able to correct the Raman spectrum for any shifts due to changes in temperature or whatever. And you can see that over 20 days, the Raman shift is better than 0.1 wave numbers. So this is a very reliable FTIR-like performance that allows you to quantify and characterize changes, very small changes in polymorphs or proteins or other such matter. The next is, how small can you go? When you look at Raman spectroscopy compared to infrared spectroscopy, Raman, you're actually getting quite a bit smaller. You're limited by the Rayleigh criterion shown in the green equations there, where for lateral resolution, we're looking at 0.61 times the wavelength of light over the numerical aperture. And for the depth axis, we're 2 times the wavelength over the NA squared. So with a 50x, your spot size is about a micron. With a 100x, you can get down to half a micron, or with immersion lenses, even smaller than that. So let's take a look at a simple Tylenol analysis, and here you can see the bands of interest for the C double bond stretch and the CH stretch, and in the software it's very easy to handle the analysis. And so we're going to put together a quick image of 0.5 microns per step, and you can see these two regions clearly shown where the distribution of these species is. So in just a minute or two, you can acquire very nice images the products of interest. If you look at a polystyrene spectrum, here's a 2-second integration and the corresponding spectrum. And then we can look at many polystyrene beads. As you may be well aware, polystyrene beads are frequently used for delivery. And in this case, we can make 30 measurements across a 4.6 micron area and be able to very nicely characterize those beads and even do confocal depth profiling to look what might be on the beads to be delivered. And here's a single image of one polystyrene bead, and you can see that it's about a little less than 2 by 2 microns. You can see that they can be well characterized. So let's look at the analysis of polymorphs. So sometimes those changes are evident in what we call the fingerprint region from 4,000 to 400 wave numbers, but more commonly for polymorphic differences, we see the changes and differences down in what we call the far infrared region, where we're looking from essentially 50 or so wave numbers up to about 800 or 900 wave numbers. And you can see on the right area of this figure that the changes that are really remarkable after the application of the pressure are shown down in that region. So let's also look at tablet analysis and putting together some quick tablet images. So in this case, we'll be interested in looking at excipient 1, excipient 2, and then acetylsalicylic acid. And we can see the 3 regions of interest up in the CH region and the carbonyl region and down in the far infrared region. And some of the tools at our disposal, you can use simple integrations. You can use multivariate quantification. You can compare full or mean spectra. That's done quickly in the software. You can do 3D cluster analysis where you'll have the Euclidean distance analysis, automated assignment of all spectra by class, and then PCA, or known as principal component analysis, where you can do an evolving factor analysis on the resultant images, and you can put together the RGB images that people traditionally are interested in from scores 1, 2, and 3. So this is what we're going to do. We're going to put the tablet underneath, and then we'll march across. The spot size is predominantly determined by the optics. So if you want a large analysis area, you use a low NA objective. If you want very fine resolution, you use high NA objectives. And obviously for very fine resolution, it takes longer to put together those images. But in any case, it's very quick and easy to look at the distribution. It's important to realize that for many tablets, the particle size they're usually 50 to 150 microns. So as you can see from just this example shown on this figure, that we can do a very nice job of imaging the tablet and looking at the distribution. You can see that the quality of the resultant spectrum is very nice. When we look at another area of the tablet, we can again see the distribution. You can click on whatever area you're interested in on the image, and the resultant spectrum is readily shown. You can also profile into the tablet. So in this case, we want to just move the tablet up into the field of view and through the field of view and see where the coding is and where it isn't for 2 different tablets. So on the image on the left, you can see as labeled where the coding is, and on the right, you can see where the coding is not. So very quick and easy to do the depth analysis. Okay, so I mentioned that fluorescence is a big issue, so how can you get rid of fluorescence? Well, Bruker has a relatively recent patent on using what we call a concave baseline correction, where if you have fluorescence due to glass, so if the glass is fluorescing, or you have even thermal heating, transient heating, you have monotonically increasing fluorescence, or many types of fluorescence. You can usually get rid of them just by modeling these broad features of fluorescence with convex functions, and then we use the concave opposing functions to strip off the fluorescence. And usually, 10 to 15 factors or curves is used to strip the fluorescence off, and it just works extraordinarily well. So let's go through a few of those examples. So the first is a coated Tylenol tablet. You can see the raw spectrum on the top, and then you can see the fluorescence removed spectrum shown in the middle, and then the reference 1064 spectrum. So the fluorescence removal really works astoundingly well, where you can see that you can hardly see any features at all in the raw spectrum, but when stripping off the fluorescence, you can actually look very nicely and observe and even quantify minority bands very nicely there. So let's look at another case that sometimes can be very interesting, where we're going to look at cellular analysis, and this is where we're going to use surface-enhanced Raman. And again, we're not interested in getting fluorescence, and just as a matter of note, you can use cheap glass slides, and they typically don't have as much fluorescence as high-quality fluorescence glass slides do. So it's cheaper and more efficient, in this case, to buy slides by the gross on eBay than to use a higher quality substrate. So remember that water immersion objectives increase the spatial resolution. So in this case, we're going to do that, and we're going to look at components of cells. This was published in ChemPhys Letters several years ago by Joseph and Aruda Yaraj, and you can see the ability to identify individual cellular components very nicely by having the signal greatly enhanced by using surface-enhanced Raman spectroscopy. And you can see that the quality of the resultant spectra are very nice. So if we want to further utilize surface-enhanced Raman spectroscopy, we can actually now include, we can do dyes and so forth, because one of the advantages of doing surface-enhanced Raman is that typical species like rhodamine, which fluoresce horribly, actually the fluorescence is quenched very significantly when you have the resonance effect. And indeed, you can go on resonance with the excitation wavelength to where the molecule or the dye molecules absorb and further enhance the signal by being on resonance. And so in this case, we're looking to do pretty close to single molecule detection. So what we're going to do very briefly, and this methodology has been patented in a few different areas. So we'll put the desired species to detect the antigen down. We'll block the system so that there's not further reaction. Then we'll apply the antibody with the corresponding dye, and then apply the silver, and then do the detection. And when you look at doing this, again, it's a 10x improvement by using being on resonance, and we're also going to have the colloid size be just right so that the Sears effect is high and the proximity is perfect, and we do that by using PVDF membranes. And again, typically several hundred molecules can be detected, and you would say, how good is that? Compared to current state-of-the-art ELISA and Western blot testing, we're about 1,000 times better for doing low detection limit analysis. Indeed, you can use isotope, different isotopic species of the dyes to achieve a multiplexed analysis. So in this case, you can see the example where we have strong or reasonable fluorescence, and then you can see with the plasmonic coupling we reduce the fluorescence. And in this case, we'll look at a typical Western blot where we have rabbit, mouse, rat, and the mixture. We have the 3 dyes, and we can do the detection all at once. And here's where you have the corresponding spectra showing the mouse, rabbit, rat, and chicken, and you can see each of these species very clearly has spectral features that are unique and allow for the quantification and detection separate from other species. So how low can you go? So this is the Western blot, and we're looking at picomoles here, and you can see that the R-squared, this is fairly early work, but you can see that the R-squareds are not bad for doing the D0 isotope and the D4 isotope of rhodamine. When we simulate or go through an ELISA 96-well plate analysis, analogous to what we did for the Western blot, you can see that we can achieve 2.1 femtograms over a 5 micron area with a good D star. And indeed, we can achieve a really good dynamic range with this method as well. And maybe the last example here is just looking at 2G gel analysis using surface enhanced solid resonance Raman, and Professor Joe Davison at the Bentley Biocenter at Purdue has published this work, we've worked closely with him over many years, and you can see that you can image the gel very nicely, and you can see the Sears signal very strongly as well. Okay, so can you look in depth with Raman? And the answer is yes, where with IR you can't. And so what you would do in the software is you just say how far down you want to go and how many steps you want to go, and off you go. So it's very easy to control and conduct either linear depth profiles or area depth profiles. And in this case, we're just going to use a confocal design where we have a slit for looking at over a large range of depths and a pinhole for looking at a narrow range of depths. And how does this work pictorially speaking? So if we're looking at the slit, we're looking over a larger area, maybe covering more than one area. And with the pinhole, we're looking over a small range of area. And again, the Rayleigh criterion holds there. The depth of field is projected into the y-axis of the detector. So to review, the wavelength is dispersed in the x-axis and the depth in the y-axis. So this is a confocal design. And you'd say, well, why not use confocal all the time and have great spatial resolution? And that has to do with taking advantage of signal. So with the slit, you obviously are going to have more signal go through. In the pinhole, you have less. So if you're not doing confocal depth profiling, you just use the slits. So there's a good example we'll look at now where, okay, we traditionally have to microtome and look across a multi-layer film very painfully sometimes on the side of these very thin films, but it can be done. But an easier way to do it, and that just works phenomenally well, is to just take the packing material in this case and look over in the depth axis from 0 to about 18 microns down. And we took 500 microns per step, and you can see the layers resolved beautifully. If I zoom in, you can see the layers easily resolved, and you can see that, well, the changes down to less than a micron are easily picked up. Okay, so the next thing I'd like to talk about for the next 15 or so minutes is the new handheld Raman spectrometer that Bruker has recently released. And we feel this is really an important tool for people to be able to use for quality control and for high throughput screening of a product coming in or going out. So the first thing to notice is that it's a Class 1 laser safe product, so that means you don't have any safety concerns. You can plug this right into your environment without any concerns at all. We also have a patent on using sequentially shifted excitation, which I'll show some examples of later, where even with pretty bad overwhelming fluorescence, we can reject the fluorescence using this method where no massaging of the data needs to be done and a beautiful spectra can be obtained. We're going to use 2 excitation lasers here. You remember I talked about using one or the other. This is a very elegant example where we're going to use 2 at the same time so that we can, because in a manufacturing world, you need to get the answers quickly and precisely. So in this case, we'll use both excitations, and the information that is most relevant for either or both of them will be used, and a very large spectral range can be obtained at the same time. The system's also smart. It has what we call IntelliTip, where we automatically have the tips that are housed onto the small spectrometer are automatically recognized. When you type in the name, it automatically will, if you don't have the right tip on there, it will inform you need to put the right one on there and detect it when it is put on there. So it's a very robust and precise optics system. It has wireless data exchange. You can run it as is, or you can hook it up to a PC. You can take high-quality spectra out of it. You can build your own libraries, and it fully is compliant with 21 CFR Part 11 requirements. So again, we have the measuring tips. There are more than one available, so they're available for virtually any application. You have a barcode scanner, so which is very handy for pharmaceutical applications. Laser release button, laser on and off, and a system on and off. And there's a battery door. They last all day on a battery charge, so that's not really a big concern. Just they're usually ready to go from the charger at the beginning of the day. So you have a bag cap and inner spacer here and a locking mechanism for the IntelliTips so that you can really have these tips work very robustly and perfect for the material you want to analyze. So the ID chip prevents the release of the laser unless the tip is connected, hence the laser class 1M laser safe rating. And even more, if the measuring tip is defined in the library for a chemical material, Bravo will inform the user that the tip needs the right tip needs to be used for the verification. Again, it's a smart tool in doing the analysis. So you can verify the sample. You'll get the green check mark and know that everything's right. You can build your own libraries. You can review results offline. You can manage the libraries very nicely, and you can change all the settings like date, time, LCD brightness, and so forth. So it's a very flexible system. So remember that sample verification workflow starts by touching the icon to get things started. So this is not a complex tool to use. Essentially, you just touch it on the screen, and away you go. And then you can scan the product to be identified, and then the batch, lot, and container numbers can be filled in or manually if you're not using a barcode identification system. And then once the sample information is accepted, then you just touch the laser icon or swipe your finger in the direction of the arrows. So this is a dynamic or active screen that you can use similar that you would use on a state-of-the-art iPhone or Samsung telephone product. So the IntelliTip now tells you which one to use, and if it's not flagging you, then you just start the measurement pressing the laser button. And then it's clearly indicated whether the product is acceptable or is rejected. The derived spectrum and library search can be reviewed. So again, if you do achieve a rejected result, it can show the spectrum, and even you can do the identification of what it actually is rather than what you thought it might be. So in the case of entries on a first screen differ in the container number only, so that, i.e. you're going to run many analysis of the same batch, you can run them sequentially without having to go back to the beginning of the measurement process. And so just the lot numbers would scale up as you go through the measuring process. So again, the verification is successful if a certain hit quality is achieved. If it's rejected, it performs the identification as we talked about. We have the ident algorithms that are state-of-the-art for doing that identification. So when you review the results, you have the ability to scale the spectrum. So in this case, you can zoom in and zoom out, looking at either one spectrum or all the spectra together, and you can move them, or shift them one above the other to get a good look at the areas of interest for doing the identification. And then to continue the verification process, you just click on the barcode icon. Okay, so when we look at the actual results here, we can see that one of the tricks for getting rid of the fluorescence is the sequentially shifted excitation. What we're doing is allowing the laser to operate at different temperatures. It's what we call actively controlled, and in this case, we'll run it at 50 milliwatts, and it's 0.8 seconds for each spectrum. This is not a slow process. It occurs very, very quickly, and allows you to now strip off the background very nicely in the spectral domain without having to do any subtractions or other manipulations or mathematics that previous methods had to use. So let's look at a wonderful example here of silicon dioxide, and we'll look at a 5-second acquisition shown above. Here where we have the 785 and 532 spectra, and you can see there's fluorescence in each case, and it's very difficult to obtain a good result, even going to 1064 looking on an FT Raman system, where if you look at with the Bravo handheld, very high quality signal to noise is achieved with no fluorescence at all, very quickly and very easily. If we go further and look at talc in polyethylene, again, the conventional dispersive Raman's spectrum looks okay signal to noise wise, but you can see that the fluorescence background there would certainly make an identification or any quantification very problematic. Where with the Bravo, you can quickly and easily obtain very nice high quality spectra very quickly using this shifted excitation method. So here's another example looking at lactose monohydrate, and again, using the SSE method, as we call it, you can generate the Raman spectrum in true spectral space, so you don't have to use derivatized results as Bruker holds a patent on doing it a previous way called seared. So this is a significant improvement over previously employed techniques, and it results in improved signal to noise compared to traditional methods to try and mitigate the fluorescence. As you can see even here, in a very quick acquisition, you can get rid of the fluorescence and have very high quality signal to noise, facilitating the identification and quantification. Next, let's look at the CH region, which is sometimes challenging if you go to the near-infrared for doing the excitation, whether you're exciting at 785 or 850 or up at 1064. And in this case, using FT Raman on a lab system, you can see that you can achieve a similar result, and on the acetaminophenol, and you can do a very nice job of obtaining high-quality data all the way up in the CH and OH region, all the way down to the far-infrared region, as we discussed previously. And when you look over the full spectral domain in this case, it really becomes very easy to differentiate between different polymorphs, as shown here on the slide. Next, let's just look at a few different cases that we see in the industry sometimes. And in this case, there is a competitor's 1064 handheld Raman unit that we kind of considered the best product out there in developing this product, and wanted to show or at least determine carefully how we stood against this product. So when we went and looked at, for example, EDTA, you can, as shown in the upper left, you can see that the Bravo spectrum is just really wonderful, very high signal to noise, no baseline artifacts, and the identification is very quickly and easily accomplished. When you look at potassium acetate on the upper right, again you can see the FT Raman literature result. You can see the 1064 handheld, and that obviously not a Bruker product, and then you can see the Bravo result. For glacial acetic acid, again, you can see that the Bravo just works tremendously well without any artifacts in the baseline. For CHAPS, same story, and then for tween 20, again, very nice spectra are obtained with very little effort or interference from fluorescence. And I think the last example I'll use is with acetaminophen again. And in this case, if you use a 1064 handheld and you compare that to the Bravo, here you can see that not only can we look at the normal fingerprint region and all the way down to the far infrared, but you can see that because we use two lasers at 785 and 850, we're able to acquire the beautiful results up in the CH and OH region without any problems. The same thing is evident when you look at cyclohexane. So high-quality spectra over a large domain is really the message for this system. And so in summary, Raman spectroscopy really provides significant benefits for analyzing pharmaceutical products at any stage of the development process, whether you're looking at clinical trial evaluation to see how the drug has affected the metabolic process, or whether you're doing at-line QC/QA analysis, or say you have a problem where there's some particle or contaminant in a product that you're not sure what it is, whether it's in a syringe or in a tablet or wherever. Raman is just marvelous for being able to do those identifications. And indeed, there are many Raman libraries now available that facilitate that identification of unknowns. And Raman can now be used very nicely for quality control for virtually any materials or compounds due to fluorescence mitigation. So this is what we believe an enabling product. This is a new capability to be able to have something that is very rugged and be able to take it out in the field or into the shipping area and be able to use it and get very high quality results without any interference from fluorescence or other problems that may be nascent to the instrument. And with that, we'll give it back to Laura for the polling question.
Great. Thank you so much, Tom, for that excellent presentation. Indeed, before we get started with the question and answer from the audience, question and answer period, we do have a polling question for all of you in the audience. And the question's up there on your screen, and you can answer by clicking directly on your screen. And as you see, the question is, how do you rate the importance of handheld Raman compared to other spectroscopic techniques, such as FT-NIR and FTIR? From not very important to somewhat important, important, very important, or extremely important. And again, to answer, you can click directly on your screen. And one more time, that just says, how do you rate the importance of a handheld Raman compared to other spectroscopic techniques, such as FTIR and FT-NIR? I'll give you just one more moment to answer, and then we'll pull up those results on the screen. Okay, so we can see the results up there, and you can see that just about a third of you said important, and another third said very important, and another 15% extremely important. So clearly there's a very strong interest in the use of handheld Raman. All right. Well, it's now time then to move to the question and answer period. So let me pull up the first question there. Okay. Tom, with the Bravo, can you get the actual peak positions of the Raman bands?
Absolutely. It's very quickly and easily read out. And again, because of the calibration of the system, the wavelength axis is very precise, and that facilitates the identification very much.
Great. Regarding raw materials QC testing, a lot of manufacturers of similar products are saying that there's a move amongst regulatory agencies to move to 100% testing of all excipients as well as API. Do you know of any evidence for a future move to this regime?
Well, those decisions are made by the FDA, and while I'm certainly familiar with many of the the actions that they're taking and have participated in some of those meetings as a bystander, I think that is certainly the goal of the FDA. They have not hid that at all. I think the Bravo is one of those tools that really facilitates that, where the analysis can now be done more rapidly and more accurately than ever before.
Okay. Is it the HQI or the p-value that is used for identification?
In this case, we're using different thresholds, so you can establish what confidence levels you want for doing the identification. But in that case, you would just you would stipulate what you want to use. So I'm not sure whether I'm answering your question or not, but you have control over what the threshold values are that are used, and stipulate what percent or what values you need to achieve. Okay.
You said that water is not a problem for Raman, but with a 1064 nanometer laser, isn't that in the NIR? And there's a little more comment here. Water is a good absorber of NIR, so surely that will reduce the energy available from the laser and decrease the signal-to-noise ratio?
Well, water, once you get to the overtones of water, so you're not in the fundamental region, and in fact you're to a many overtone region when you're up at 1064, and even more so when you're at 785 and 850. So the absorbance is horribly low, so water's not interfering at all. The decrease in signal is negligible and challenging to detect at all, really. Remember, water is a C2V molecule, which by selection rules means it has a very, very small Raman cross-section. And because the absorptivity of overtones is so low as well, water is not going to bother you at all.
Does the Centara have SSE technique for fluorescence rejection?
Good question. It does not right now.
Okay. Next one.
It uses a fluorescence removal technique, what we call the concave baseline correction, which works very well. But certainly, the Bravo is employing new technology.
Great. The next one's sort of a complex question here about what you can measure and in what context. So the person says, plastic bags and wrappers are achievable. Is this for transparent bags only, or will it work on opaque bags too? Additionally, are any of your products capable of analyzing through a paper sac?
Okay, so those are all related questions. So if you're looking to go through packaging material, which is many times transparent to visible or near infrared excitation light, that's fine, and you can characterize the material inside the packaging nicely. If you're looking at trying to look through something that's highly scattering like paper or other opaque materials, obviously you're not going to be able to get significant laser energy back to the detector. So in those cases, you'd have to use some other tool.
Okay, we have a couple questions about Raman libraries. How long does it take to develop a Raman library if you need to develop your own? And how many scans are required to create a library entry?
So the number of scans is dependent on how good your sample scatters light. So if you have a strong Raman signal, it can take as little as a few seconds, and where it may take longer to actually type the name in than it would to actually collect the spectrum. If your sample's weakly scattering, then you would have to, at that point, move in and scan maybe for a minute. But the acquisitions are fast.
Excellent. Is this meant to be an alternate ID test at incoming QA inspection, or as a replacement to current methods?
I think you have to transition. So whatever method you're using now, you're probably reasonably happy with, at least in being compliant. And so I would use both to show the capabilities of the new technology. The FDA has, according to their protocols, actually supports transitioning to newer and more useful technology, such as something like the Bravo. So you would use both for a short period of time and then transition over to the new technology. And the FDA, again, is fully supportive of that methodology.
So I think you just answered this, but just to be sure, how are companies typically submitting Raman ID tests to the FDA?
You would submit the ID test the same way you always would. So for the details of that, you should go onto the FDA website. And I'm not going to paraphrase their documents at this point. It doesn't make any sense. You should go directly to the source for how to submit those certificates. Mm-hmm.
Great. Will this new instrument distinguish between polysorbate 20 and polysorbate 80?
I haven't tried it, but if they're spectrally different, it will. I know that's kind of a quaint answer, but if you want to send me your polysorbate samples, I'll turn them around right away and see if we can differentiate them nicely. So, and that is true for anyone listening. We're happy to make trials for you, and if you're interested in actually trying it out, you can let me or your local sales rep know, and we're happy to come out and visit with you and let you run some of your samples and see how they look to you.
Great. On that point, here's another question about samples. Can Raman differentiate colors? For example, Opadri is an excipient used in production but has various colors. Can Raman be used for an identification test of this excipient?
My experience has been is that down where those color pigments actually have absorptions, they're organometallics. So down in the far infrared region, usually you can differentiate them.
Great. Can you say a few words about sample prep? Can you sample finished tablets directly?
Whether you're doing micro Raman or you're using the handheld Raman, the intent is not to have to modify the tablet at all. It's undesirable to do that.
And there's a related question, which I think you addressed with dealing with the plastic bags, but the question is, can this test be performed without removing the sample from the shipping case?
Well, if the shipping case is cardboard, you'll have to take it out of the case, where I can't shine the laser light through it. But if it's something that is transparent, of course, you should not remove it from its shipping case. If it's just clear plastic, the Bravo analyzer can look very nicely through the plastic. And as a matter of fact, so can the Centera Raman microscope with a 10x objective. You can look through 2 polyethylene bags and identify an active or whatever very quickly and easily.
Great. And so a closely related question, what is the penetration depth of the Bravo laser, and can you measure through a thick glass container or HDPE? Yes,
so you can look through thick containers. The depth of penetration is again dependent on the material you're looking at, but typically you're looking at 100 microns or so typically. Sometimes a little more, sometimes a little less. Again, the scattering properties and refracting properties of the sample are relevant in that case.
In this handheld Raman, is there any fiber optic part?
The current implementation does not use a fiber optic port, and the main reason for that, and I'm not the development engineer, I'll just speak for them in this case, is that, well, the spectrometer is so small, there's no point in bringing the signal back to somewhere else. The intent is that this thing is small enough and easy enough to handle, you can take the whole thing right to what you want to analyze. So there's minimal facility for using a fiber optic cable in that case.
What type of grading is used in the Bravo unit?
I'm not going to give out that information.
Okay. Is it recommended to challenge the robustness of the method, for example, in terms of humidity and light?
Humidity, again, is just going to test how good the instrument is if it's in a hot, humid area. We've done all that kind of lifetime testing, so it'll work. It comes with a good warranty. As far as spectrally speaking, as I said, water's not an issue, so it should work great.
Great. Do we have to build the PLS model for quantitative analysis using Opus for Bravo data?
That's the best way to do it because you have all the tools. You have a full PC-based workshop to handle the analysis. I would say that's the best way to do it.
Do you need to create the library and use ident in the same way that is done with the Bruker FT-NIR MPA? And this question relates
to the Bravo.
Way.
You can do it that way. As I mentioned in one of the slides, there are many different analysis algorithms available.
And do you supply any sort of library with the Bravo?
There are libraries that are provided for pharmaceutical analysis, and again, it's easy to add your compounds to the database, so absolutely.
What is the spectral resolution of Bravo?
I probably don't want to give that answer, actually. So maybe offline I'd be happy to answer that question, but not in this forum.
Okay. Here's a question. I'm not quite sure I understand it, but I'll read it to you as written. How many wavelengths does Bravo laser source can switch?
It doesn't switch. It uses simultaneously 2 different excitation lasers.
Excellent. How often can Raman spectrometer of handmade, I think they mean handheld, be used in protein gel bands constitution?
I'm not sure I get the question, but it can be, there's no limit. The system is intended to last for a very, very long time, lifetime-wise. And so in doing protein gel band analysis, maybe you're concerned about the gel breaking down. It's intended, again, that the Raman analysis and the surface-enhanced Raman analysis, in this case, be non-destructive. So whatever the normal decay time would be of the gel pack, we would expect the same thing here.
Great. Do you use the same number of scans for your library spectra and for the sample being tested?
It's not important because, remember, it's the signal to noise that's important. So the library you want to build up so that there's little to no noise. But when you're actually doing the analysis of the sample, in that case, maybe you scan for a few seconds and you have achieved the result that is very secure.
Is there a dedicated tablet holder that comes with this or is available with it?
I believe there's a very nice dedicated tablet holder so that you can very quickly and easily run through tablets.
Excellent. Does Raman work well with functionally coated tablets and identifying the type of coating and thickness?
Yeah, I think that confocal Raman is just ideal for that. So now I can use a confocal design where I'm looking at the surface, and I get only the surface actually in the confocal. You are explicitly rejecting that signal from below the surface, and then you just march down and be able to collect the data to characterize not only the coating but also below the coating, and to look at the gradient and see whether the coating is penetrating into the tablet, which may or may not be important for dissolution studies. So again, confocal Raman is just a marvelous tool for looking at those problems.
Can the library developed on other vendors' instruments be transferred to Bravo or vice versa?
Almost any library you have can be used directly with the Bravo. That's very easily done. As far as taking them out, that's not my problem to go to other instruments.
Sure. Can you export the spectra for analysis in additional software packages?
Sure.
Okay, great. How long does it take to get a Raman image using the microscope?
Depends on the signal to noise, and this is true of virtually any system. There's a lot of misinformation in our field where people will say, well, I can collect 5,000 spectra per second using this imaging mechanism. Well, it's sometimes ridiculous. If the signal to noise is poor on that system, you're signal limited. So if it takes a second at each point to get a good spectrum and you want to collect 5,000 spectra, it's 5,000 seconds to acquire the data. If the signal to noise is strong, where you can collect many spectra per second, then the time will be much shorter. So usually you're looking at being limited by the system more than by the instrument.
Great. Maybe we'll just do one or two more questions. Has Bruker Optics considered optimizing its new handheld Raman systems for trace detection in security applications?
We have considered it, and we'll get back with you. That's maybe, Nicholas, you should just send me an email offline, and we can talk about that further. Fantastic.
All right. Well, I think it's time to wrap up. Thank you so much, Tom, for your excellent presentation today and for answering all those questions.
Yeah, maybe those that did not get your questions answered, I see there are a handful still left. You can, we'll address those offline for you, so we'll still answer those for you later.
Fantastic. I'd also like to thank all of you in the audience for participating today and for asking all those good questions. And I'd like to extend our thanks to our sponsor, Bruker Optics, for making today's webcast possible. This webcast will be available for on-demand viewing through March of next year. You'll receive an email from Spectroscopy alerting you when the webcast is available for replay. We invite you to forward that announcement to your colleagues who might have missed today's live event. We hope to see you all next time. Goodbye.